Hey, what you got there?
Legos. Look, I made a house.
Oh, that's cool. Hey, look. I made a DNA molecule.
Huh? That's just a block.
No, it's a DNA molecule.
What's PNA?
DNA is the genetic material found in all cells. Each strand of the double helix is composed of nucleotides, which each contain a phosphate, a 5-carbon sugar, and a nitrogenous base. DNA is extended during replication or synthesis by the attack of the 3' hydroxyl of the sugar on the alpha phosphate of deoxynucleotides in solution, creating a phosphodiester bond.
Huh?
Okay, it's like this. There are 4 different nucleotides in DNA, and we've got 4 different colors of Legos right here. In Legos, you can make a really long stack by putting the top of one into the bottom of the other. See?
Mm-hmm.
In DNA, it's almost the exact same thing. You can make a really long chain of DNA molecules like this.
Okay. Can I just have my Legos back?
Scientists have known for over 100 years that DNA is the major genetic material in most organisms, but they didn't have a reliable, efficient method for sequencing base pairs. This was the case until 1977, when Frederick Sanger developed a method now known as Sanger sequencing.
You start with a single-stranded piece of DNA to be sequenced, and add nucleotides and DNA polymerase, which will attempt to copy the DNA strand by matching nucleotides to the template strand. However, at the same time, you add a small number of nucleotides that are missing both the hydroxyl group in the 3' position on the sugar and are fluorescently labeled in different colors according to which base they have. The polymerase starts at the beginning of the strand and adds complementary nucleotides until it uses one of the modified nucleotides by chance, which ends the strand because there's nothing for the next nucleotide to attach to. This results in a short strand of DNA that is fluorescently labeled on the end. You can then separate fragments by size and determine the sequence of the original DNA strand by reading the fluorescent colors from smallest fragment to largest.
Huh?
In terms of the Lego analogy, it's like you have a long chain of nucleotides. That's the template strand, and you match the corresponding color next to it. You keep adding the Legos, but at some point, no more Legos can be stacked. Once you have all your Lego chains of different sizes, they can be ordered according to their length. Real Sanger sequencing is a little different than Legos. All the DNTPs are different shapes, while all the Legos are the same shape. Also, hydrogen bonds form between the bases on the complementary strands, but when you use Legos, nothing is there to hold the 2 strands together. Finally, in Legos, you can see all the colors in the chain, but in Sanger sequencing, you can only see the color of the last one due to the phosphofluorescent label.
Frederick Sanger was a pioneer in the field of DNA sequencing, which earned him his second Nobel Prize in 1980. His work paved the way for many other methods, such as high-throughput sequencing, which breaks up the genome into small fragments for sequencing, then overlaps the fragments to show the sequence of the entire genome, or nanopore sequencing, which uses electrophoresis and electric currents to determine the sequence. DNA sequencing has had a profound impact on the world. In terms of research, it has revolutionized the way we study genetic disorders, the molecular basis of different diseases such as cancer, and to identify drug targets. It can be used as forensic evidence and to determine paternity. As DNA sequencing has become more and more inexpensive, it has also become accessible to the public. Today, people can take direct-to-consumer genetic tests to learn about their ancestry and health-related information. However, there are some potential concerns surrounding the ability to sequence DNA, as it could be used as a form of discrimination. For example, if a person knowingly has a genetic predisposition for a certain disease, an employer could refuse to hire them, or an insurance company could refuse to cover them. Additionally, there is the potential for DNA information to be used for eugenic purposes.
Wait, what's eugenics?
So, you see, eugenics is a...
Stop. That's a story for another day.
Overall, however, Sanger sequencing and DNA sequencing in general have been very beneficial to both the scientific and public worlds.
So Sanger sequencing is pretty interesting, right?
Yeah, I'm gonna be a scientist when I grow up.
Wow, that's really cool.